So I would like to say that first of all it was my great pleasure to see the people coming, as maybe not all of them are members of this chemical society unit, but anyway. And today, or maybe I can talk this way.
I think it's better.
Okay, so I will try with it. So we'll talk about the so-called super resolution microscopy. Occasionally it is also named nanoscopy, but I don't like this term, so it's better to say that it is super resolution, and I will explain in details what we are talking about. So what for Nobel Prize in chemistry was awarded. So actually, if we look deep into history, there were several Nobel Prizes awarded for the development in microscopy. This is not the last one. The last one was announced last year, but this will be a separate topic. So this is the formal thing, that this award was given to 3 persons, Eric Betzek, Stefan Hell, and William Merner. And this is the official announcement for the development of the super resolved fluorescent microscopy. So when we are talking about so-called super resolution microscopy, we'll talk exclusively about fluorescent microscopy, because no other mode in microscopy allows us to break the diffraction limit. So the invention, the history. The history starts from the 19th century, when Ernst Abbe formulated the limit of the resolution of the light microscope that depends exclusively on the wavelength of light that we are using. And for decades, people tried to use shorter and shorter wavelength of visible light. Then people went to UV light, but it was fairly unsuccessful. And then the 2 separate principles that are rewarded, they were introduced at the end of 20th century. So the overall story lasts for more than 100 years of continuous efforts of different people. And it became possible only with special achievements in computer technologies and in laser technologies. So both things were involved. So one is so-called stimulated emission depletion that was invented by Stefan Hell and developed by him. This will be the first part of our talk. And another part, so this is just from the official side of the Nobel Committee. And the other part will be about the development on so-called stochastic approach to the super resolution microscopy, where Eric Betzek partially was involved and Merner was not involved at all. So far, we have 2 different principles how to break through the diffraction barrier. One approach is using STED, stimulated emission depletion. That means scanning of the specimen with tiny excitation volume. And the further development of this is resolved. It is closely resembles STED, and I will talk about it later. Reversible, saturable, optical linear fluorescence transitions. And another approach is totally stochastic, based on the idea of random excitation of a low percentage of molecules in the specimen. And there are 2 major approaches: STORM, stochastic optical reconstruction microscopy, and PALM, photoactivated localization microscopy. So both approaches use the same principle, but PALM is more related to the observation of living cells, while STORM is related to the study of only fixed specimens. So first of all, let's start with Stefan Hell. I just want to say a few words about his biography. Right now, he is a director of the institute, head of a big department, But when he decided how to make the STED microscope, he couldn't find even a position in Germany. So he went to Finland as a postdoc, and in small Finnish university he developed this instrument. But after the implementation of the STED microscope appeared to be successful by the end of 20th century, he was invited back to Germany. So, and he made a lot of other minor inventions in microscopy as well, besides STED. So it was invented in 1994, and nobody could say that it was feasible, that it was realistic, nothing. So this was done in Finland and realized in 1999, and then immediately he was invited back to Germany for the professor position. As a principle, this is deviation of STED. I will talk a little bit later. So now let's go a little bit deeper and see what is the principle of STED-stimulated emission depletion microscopy. The idea is very simple if you know how the molecules work and what does fluorescent means. So we have absorption process initially. Absorption is very rapid process. It takes approximately 1 femtosecond. That means 10 in minus 15th degree, very fast process. Then for the fluorescence, that means emission of another photon, it is a relatively slow process, and usually it takes about 1 nanosecond. For us, 1 nanosecond is vanishingly short time, but for the molecules it's relatively long time. So the idea is that there might be another way how to manipulate with emission. And Stefan Hell decided that we can deplete the major part of the fluorescence by another excitation. And that means stimulated emission. So the overall idea is the follows. We have 2 lasers. One is used for excitation, and it excites normally, as in confocal microscopy, the relatively small volume that represents the airy disk volume. This is standard. And simultaneously, there is another depleting laser with another wavelength that gives much more powerful pulse, and these pulses should be very short, because the idea is that using another wavelength, we stimulate emission in a very short period of time. So instead of waiting for nanoseconds, this population of molecules is depleted within few picoseconds. Actually dozens of picoseconds, but still below 1 nanosecond. Then, if we start our observation not at the time of irradiation, but let's say 1 nanosecond after the pulse, we open the shutter of the camera. What we will see? We will see only the fluorescence that will come. The fluorescence will continue for many nanoseconds. That will come from all the molecules that were not depleted from the fluorescence by stimulation emission. So this is the idea of the temporal shift between 2 states. And this is in time. And then it comes in space. In space, the approach is the follows. We have the major beam that excites, as I mentioned, the entire airy disk, and another beam that is toroidal beam. So it is defocused compared to the first one. It just excites, no, it does not excite, it depletes the signal from the volume around the center of the airy disk. So we have the airy disk excited and then depletion around the center. And only the small area in the center of the airy disk is not depleted. Actually, it is statistics, of course, So we can say that we deplete this area to, let's say, 99.99%. We cannot deplete it completely, it's always incomplete. But here in the center, the energy of depleting laser is reduced. So far, a significant portion of molecules survived through the second pulse, and they are emitting their photons as usual. And as a result, the profile of the volume might be very, very narrow. And the formula is given here. It is the estimation of the diameter of the volume that could be excited, specifically by using this double laser pulse. It means that we have now another tool that is similar to the laser confocal microscope, but it is working in slightly different way. So if in the confocal microscope we can see the airy disc of this size, approximately. This is XZ projection. It is not an XY plane, it is X and Z along the axis. Here we can reduce the excited volume by this toroidal, so here it is again XZ projection, so along the axis it is not exactly toroidal, it's more complex. But this is depleting beam, and so only the hot spot here is excited. So this is the real tool that allows us to excite a very, very small volume that is in diameter, let's say 30 to 40 nanometers. Now it is achieved even less. This is a relatively old picture, but it illustrates the idea. So with modern technology we can reduce this volume twice compared to what is shown here. About 40 nanometers is already achieved. And then we can scan the entire area in XY with very small steps and generate a super resolution picture. So this is the idea of STED microscopy, and this is the summary illustrating what I just described. So by increasing the depletion, the power of the depleting laser, energy of depletion, we reduce the excited volume, of course to the reasonable minimum. The main problem is that since we are decreasing the excited area, the steps in our scanner should be tiny. We cannot go with the similar steps as in regular confocal microscopy. So the steps here will be, if we are thinking about the resolution of, let's say, 40 nanometers, the steps will be 20 nanometers. 80 nanometers, roughly, it's an estimation, will give us just 40 nanometers step. So it is easy to understand that the process will be relatively slow, and if we wish to cover the area that is a significant portion of our normal field of view, that is under 100x objective, usually more than 100 microns, but let's assume 50 by 50 microns with 20 nanometer steps. That means 50 steps per micron. So it is enormous. And that's why this technology in reality allows to work with a fixed specimen and to take pictures from relatively small areas. Typically it is not more than 20 to 20 microns. But it was Stefan Hell, so he went further. These are the pictures that you can see, just demonstrating what is the power of the STED microscopy compared to the standard confocal. Virus particles, 40 nanometers in diameter, they are fluorescent particles, and here you can see the relatively big point spread function of individual particles in confocal, and this is instead microscopy. Another picture just showing more real portion. This is a receptor of the potential channel on neurons, and we can see how it looks like in the regular confocal microscopy, and here we can see the minimal dots that are about 40 nanometers in diameter, individual signals. And we can see where the dots are clustered, just compare this and this, and where they are separate from each other. And even more than that, it is possible to achieve the 3D picture. Of course, it is limited, but this is on the top of the capabilities of STED. So it is portion of the dendrite, individual optical sections were taken and then summed. So this is also from the health lab. The paper was published just 10 years ago. And the pixel size here, the pixel size means the scanning distance, just 29 nanometers. That means the overall resolution here is about 60 nanometers in XY, and in Z it is also less than 250 nanometers. Now, can we improve our pictures? Yes, we can if we add another option that is regulated time delay for the readout. Because fluorescence is decaying process. We are not talking about individual molecule that is fluorescent at a constant level. So if we make the gated step, and this means that we are not starting at the very beginning, we are waiting for 1 or even 2 nanoseconds after the end of the pulse, of the stimulating pulse. Then we can achieve better spatial resolution. So here the delay is 3 nanoseconds, and this is so-called continuous wave. That means immediately we start readout, and again comparison with the confocal microscope, and this you see the spots are becoming lower and lower in diameter, but unfortunately the brightness of these spots is also decreasing. So for the model object, that's fine, but for the real microscopy, it might be a problem because of the relatively low signal. And this will be just several slides showing the modern capabilities of STED microscope. This is the nucleus and nuclear pores, and with gated STED, we can increase the resolution and resolve the pictures of the individual pores at a distance of less than 100 nanometers and down even to 50 nanometers. This means gated detection. Another option introduced later by Leica, and Leica are exclusive distributor of STED technology. So they signed the special agreement with Stefan Held before he became Nobel laureate, of course, so it was cheaper for Leica, and so the major things instead go through them now. That means we can manipulate with depleting beam, changing its shape. We can achieve the minimal diameter in XY given on top. That means it will be very, very narrow in XY, but of a relatively big length along the Z-axis. And we can minimize along the Z-axis by just shifting one beam in relation to another beam. And it will result in increased area in XY, but significantly decreasing the depth of excited volume. That means we can balance the resolution in all 3 dimensions. Because one of the major problems, if you are thinking about 3D microscopy, is relatively low resolution of the microscope along Z-axis. It is on average 3 times less than in XY, and here specifically it might be a big problem. So this is just to show how we can manipulate with the shift in the depleting beam. And this is the balanced STED mode, what they call the balanced STED mode. Just a picture showing how it works. One picture, so we can now use a STED with different colors, with the development of special objectives with very, very high chromatic aberration correction. So this is important instead. and we can achieve this resolution. Here is another idea how to decrease the depth of excited volume. I'm not going into the details. It is one of the latest publications from Stefan Hell laboratory. Again, it is from his lab. So putting specific pattern for the wave front of depleting beam, we can reduce the depth of excited volume. And these are just some pictures. Mitochondria reconstructed in 3D with a STED technology. From the same cell, bundles of microfilaments around the same mitochondria. Just for you to get an impression, the size, this is about 10 microns. And Vimentin intermediate filaments surrounding all the cage. Now, what are the limitations? As I told you, the major obstacle in using STED is very slow scanning as a result of very high resolution. So this is here. And the resolution also is not very high if we compare to the standard. It is significantly higher, but 2-3 times or even more, but not 10 times. So another option is so-called resolved microscopy. I'm not going to read all these texts because of the lack of time, but I will show the principle. The idea is, instead of making single point source, we make multiple sources. And this is also another invention from Stefan Hell and his group. So we put the graticule in front of the lens. And we have now at the focal plane of the objective such a picture. It is diffraction pattern from the grid. And by using depleting beam through this grid, putting the light from the depletion beam through the grid, we have such a pattern of light, and we can deplete significantly our light that is coming through as a wide field illumination. So the idea is, instead of putting the focused beam and depleting around it, we put defocused beam for the entire field of view. But the second laser pulse is going through the diffraction grid, and it is depleting many points of the specimen except the central hot points. What it gives to us? Now, for getting a relatively big picture, we don't need to scan the entire area. We just need to scan at the distance from one maximum to another maximum. That is relatively small. And it means, and the same in another direction. So instead of millions of scanning points, we have just hundreds of them. And this accelerates the readout rate of the system. So these are the pictures that were obtained from live cells. So now the time for taking the picture is going down from many minutes to few seconds, because we have to take only about 100 of consecutive images. And this is, of course, much faster process. Keratin filaments, this is wide field, and this is resolved microscopy, and the resolution is increased approximately 4-fold. It is below 80 nanometers here. And this is the enlargement of the wide field area, and the same picture obtained using the resolved technology. Here you can see one bundle of filaments, another, and the third one. So these are what we have, and also in 2 colors it could be done in the same way, and I just want to show these pictures for you to understand how it looks like. Again, it is everything from Hell's laboratory. That's where the technology of resolved is now, the acquisition time is going down to few seconds, so it is feasible to work with living cells, of course, for relatively slow processes. And the spatial resolution is about 70 nanometers. I think if Hell will be not German but American, they will put the numbers 1.5 times less. But he is always underestimating his achievements. Now another approach, the second part of the story. Stochastic super-resolution microscopy, storm and palm microscopy. So the idea of the resolution limit here is the follows. When we look at the individual molecules, we see the airy disk of each molecule. So whenever these airy disks are coming close, they fuse for us and we see one and the same spot. However, there is technology now, it is computer-based technology exclusively, when we can reconstruct the position of the individual molecule with very high precision, assuming that there is only one molecule in in a relatively big circumference. So we are not talking about the molecules that are closer together. We are talking about the reconstruction of the position of the individual molecule. This is the normal situation and this is the reconstruction. So how it works? This is the principle. If we assume that we have several molecules excited and they give us fluorescent signal, and we measure this fluorescent signal, normally accumulating this signal for many nanoseconds, typically for milliseconds, and this is Pseudo's 3D picture showing the intensity and the XY scale, the pixels of the camera. If we look in the enlargement, we can see that each pixel receives different amount of photons, different number of photons. And from these numbers, we can reconstruct the position. This is mathematical procedure. I'm not going into details and I'm not going to kill you with the huge equations, but we can determine if the number of photons is significantly big, we can determine this center with minimal error. The larger is the photon flux, of course keeping in mind that the molecule is not oscillating. That's not true. At the room temperature they are oscillating. So the accuracy of such measurements is still limited, just from the thermodynamic point of view. But this is the counts, and we can see if we have the counts at peak pixel. So here we see there is one pixel that achieves maximal number of photons, and its neighbors achieve less number of photons. But calculating the ratio between the density of photons on the neighboring pixels, we can determine this center in more details. So this is the idea of the method, and the accuracy of the detection is proportional to the square root of the number of photons, but if we do not take into account the thermal noise. So let's just sacrifice that there is no thermal noise for simplicity. And then with 1,000 of photons, we increase the resolution roughly by 30 times compared to the normal limit that is given here. So this is the idea how it can be calculated. And there were several groups of researchers working on these calculations. Nobody of them were awarded Nobel Prize, so they are just in the shade. This is the accuracy how you can calculate it. And now I'm going to say about the implementation of these calculations. So we can, first step that was important, and that was not maybe recognized as a real breakthrough, but it was that at the beginning of 21st century, the high accuracy of calculations. Now, we can detect the signal from individual molecules. To make sure that we are detecting the signal from individual molecules in the early years was the picture that I showed. The fluorescence is nearly constant and then suddenly drops down to the 0 level. But when we are talking about multiple molecules, we need to ensure that they are individuals. So, setting properly the camera, recovering the centers of the airy discs for each molecule, and sequentially taking multiple images and overlaying them. So the typical number is 30,000 of sequential images of one and the same area. So it's not very fast process. And I will show one of the pioneers of this method and was Xiaowei Zhang. She is working at Harvard University, and her group achieved the top resolution in the light microscopy that I will show in a few frames next. So the first approach is so-called storm stochastic optical reconstruction microscopy. So I don't need to put microscopy here, but for clarity I added. So there are two lasers that work by, for producing the regulated fluorescence. And here how it works. For each pulse, when we take one picture, very low number of molecules are excited. And this is a special technical thing. I'm not going into all the details. So one frame looks, actually this is not one frame, this is already about 100 of frames. In one frame you have very few dots. Then summarizing, it is continuous read, that means there is short pulse of excitation, then the frame is taken by the camera and going to the computer. The positions of all molecules in the frame are calculated by the computer, all these centroid calculations. Then the next frame is, the next pulse of excitation goes to the same area, the second frame is taken, and so on. So finally, here it is about 700,000 images, we can see such a picture. And this is just fluorescent image of the same area that we can easily get by wide field microscopy. So the equivalent pixel size of the camera is relatively large, but since we have only dots, the position of each dot might be restored with very, very high precision. So just a few pictures to show how it works and the resolution that was achieved. Here it is wide field microscopy and storm microscopy. We can see the resolution going down to 37 nanometers here. This is another picture of double color storm. We see individual microtubules. They are 24 nanometers in diameter, and apparent diameter here is less than 100 nanometers. And collecting vesicles that are approximately 200 nanometers in size. That means we can see them as dots under conventional microscopy, but here we can see them as real vesicles. Now the same question, we achieve very high resolution in XY, what about Z projection? To increase the resolution in Z projection appears to be tricky, and for that we need different approaches, and different approaches were used. One is so-called astigmatic imaging, when we put the lens, the cylindric lens, that gives us a astigmatic image of everything except the portion in the focus, in focus, exactly. Or P-plane imaging with 2 CCD cameras and another way. So the best choice is just putting cylindrical lens. And to further increase signal-to-noise ratio and to get more light, the double objective principle was realized, and this is from Zhang laboratory, this microscope. So we have such images where we can distinguish that molecules which each molecule which is in focus gives exactly the circular image. The molecule slightly shifted from the focal plane becomes elliptical. Her image, the image of this molecule, becomes elliptical. And it becomes elliptical, and we can see them and just withdraw them from calculations. Here the arrows show these ellipses. And we can achieve, so they achieve the resolution by slow summing the images of 10 nanometers in XY and not more than 20 nanometers along the Z-axis. So this is the picture where along the Z-axis position of microtubules was scaled. So this is fixed, of course, specimen, it's not live. And to take this series of images requires several hours. So they don't put it explicitly, but the estimation is about hours. And the scale represents the resolution along Z-axis. Overall scale is 600 nanometers. And you can see the microtubules at different positions. And this is, I think, the most impressive picture. It was published in 2012 by Jean Leb. Here is a microfilament system at the edge of fibroblast. And we can see this system. It was scanned along Z-axis. The ventral surface, the surface that is sitting on the cover slip, and the dorsal surface that is above. The distance between them is less than 1 micron. The resolution here is about 10 nanometers in XY. And you see the big difference in organization of actin filaments on the two surfaces. So each microfilament system is sitting just beneath the plasma membrane at a distance of few nanometers from the membrane. And this is the big difference that was observed for the first time. Conventional electron microscopy does not allow you to get such a resolution because in microscopy, for 3D, we need restoration of the image. It could be done, but it is more difficult. Now the third approach that is very similar to the STORM, but it uses the photoactivated proteins and could be utilized for living cells, where Eric Betzig was involved, found photoactivated localization microscopy. So the principle is exactly the same as I told you. We excite individual proteins, then determine their position with maximal precision and summarize thousands of pictures. But the situation is a little bit more complex when we are using standard palm, because there is activation first to activate small fraction of fluorescent molecules. One laser pulse, usually violet. Then excitation and then bleaching for the same molecules not to be activated anymore. Because for random activation, we prefer to bleach the molecule that were activated. And one frame after another, we can assemble the image. So these are two variants of the modern method because the palm technology relies solely on the properties of photoactivatable protein that you have. So you cannot buy the palm microscope. It is totally useless unless you have these proteins in your hands and can manipulate with them, putting them, transfecting cells with them, and getting stable expression at appropriate level. And these are special photo-switching proteins or photoconversible protein that can switch on and off. And this is a huge industry now in the development of new proteins. So in the last 5 or 6 years, I think many of these proteins were developed, and now this becomes more and more common. And this is the comparison, how the pictures look like at the conventional fluorescent microscopy compared to the palm approach, and in these focal adhesions now we can see numerous clusters of paxillin. I think, yeah, it's paxillin molecules inside the single focal adhesion. So if we compare now palm and resolved microscopy, so these two can compete somehow in the evaluation of living cells. This is the brief summary. Resolved allows to achieve 60 to 80 nanometer resolution, and the time needed for resolved might be as low as 2 to 4 seconds. For palm microscopy, still, despite we have very fast cameras, and the readout rate is more than 600 megahertz now, still it is about 20 seconds, because the problem, so far the problem with the living cells is still existing with the palm technology. Only something relatively stable we can really see there. And this is where I would like to stop, and maybe you will have some questions, and I am ready to answer them. Thank you.